next speaker is Qingsheng Hui. Is that correct? Yeah. And he's coming from Shandong University. He's going to speak about exploiting the novel potential of E. coli in industrialized microorganisms.
My talk is about exploiting the novel potential of E. coli. Which one is already an industrialized microorganism? E. coli is actually a model for basic research. Exists everywhere, and the physiological background is well known. So a lot of genetic method was developed based on this strain. It is now almost a model for synthetic biology. E. coli has become an important protein factory since the last 80 to 90s because this strain can easily express proteins, and the fermentation process is easy also. It can reach a high cell mass. So a lot of pharmaceutical proteins such as beta interferon, EGF, and also EPO has already been produced in this strain. Now this E. coli has become a potential chemical factories because it's easy to grow even in a minimal medium, and it can use many kinds of carbon sources such as xylose, arabinose, as well as fatty acid. Metabolic pathways of many chemicals has been set up in these strains, such as some acid, lactic acid, acetic acid, and succinate. And by metabolic engineering, a lot of unrelated chemicals are also produced in this strain, such as propanediol and also PHA, which E. coli, as we know, is a non-native PHA producer. Only by metabolic engineering, PHA can be produced in E. coli. We have to mention that this biomass for production of chemicals should be cheaper. For example, we should use can-molase, which is whey, or hemicellulose hydrolysate. With this cheaper biomass, substrate mixture was happened. For example, there's glucose, arabinose, and fatty acid also exist in the picture. So by metabolic approaches to develop a base strain for metabolic engineering or for industrialized strain, regional metabolic engineering should be done, which based on the available information, set up this metabolic pathway by simply gene cloning. Inverse metabolic engineering, which was also done based on the phenotype of the strain, analyze the genetic background of this phenotype, and transfer this genotype to the production strain. And random engineering strategy also should be done. This random engineer strategies are based on the random mutagenesis, which include evolutionary engineering, transcriptome mutagenesis, and gene overexpression library, and global transcription machinery engineering. All this could be set up for a base strain. However, to develop a system strain or to develop a system method for this whole improvement, a system metabolic engineering should be performed. In this case, you should first set up a base strain by what we mentioned, such metabolic engineering, and then we should do this large-scale genome-wide in silico metabolic analysis, and in third round, integrated bioprocess should be done. However, to develop industrial E. coli, not only the metabolic pathway should be set up, but also the bacteria itself needs to be improved. This includes to improve substrate utilization, a wide range of substrates, including xylose, aminose, can be consumed simultaneously. And second, you should maximize the biomass and the product accumulation with a stress resistance capability. Third, we should enable the cheap and simple fermentation process with a suitable induction system. And last, we should enable the strain to have an efficient product recovery process. So to improve the substrate utilization capability, we analyze the metabolism of the E. coli. We decide to knock out the PTSG, the importer of glucose system. We knock out GLK and to regulate the GLP system to let this sugar transferred through this GLP transporter, but not PTSG system. In this case, we realize that the xylose and glucose consumed simultaneously, although a little bit the xylose was a little bit slower compared to glucose, but basically they are consumed simultaneously. Second, we should also think about fatty acid. This fatty acid is also exists in some case in the medium, so we should incorporate fatty acid. Normally, the fatty acid cannot be used in the presence of glucose. So we overexpress BAD, actually a fatty acid acyl-CoA synthetase. Overexpression of this gene can lead to the E. coli to use fatty acid with chain length lower than 12 in the minimal medium. Normally, E. coli can only using this fatty acid with chain length lower than 12. But overexpression of this BAD gene leading to the remove of FAD-R repression to the beta oxidation operon. Then this all the genes in the beta oxidation operon can be expressed, even in the minimal medium supplied only with carbon sources or fatty acids with chain length lower than 12. So we further incubate this E. coli in the presence of fatty acid at different concentration. We found that the E. coli harbored FAD gene can resistant high concentration of fatty acid. This means that this E. coli can grow in a little higher concentration of fatty acid. So we combine these two ideas. We knock out PTSG and FAD-R. Then we found that the E. coli can simultaneously incorporate fatty acid and glucose. In this case, this E. coli can incorporate fatty acid, various sugars simultaneously as a substrate. Also, this fatty acid has a little bit lag phase compared to the glucose in this case. So, to improve this the capability of E. coli, not only the biomass incorporation should be considered, but also this is actually this RT-PCR analysis of the metabolic pathway in the beta oxidation of E. coli in the presence of glucose, that the proteins in the beta oxidation pathway was also expressed. This is RT-PCR result to confirm that result. To not only to consider substrate utilization, we should also consider of acetic acid and propionic acid, which is normally exists in the medium. So we also regulated this expression of PRPE and AKA genes. A regulation of PRPE by a strong promoter can lead to the more fast assimilation of propionic acid, and SKA can assimilate acetic acid. So by rational designing, we developed a base strain. This base strain, sugar such as glucose, aminose, xylose can be consumed simultaneously. Second, fatty acid, even short chain length fatty acid can be consumed simultaneously. Acetic acid, propionic acid can be also incorporated more efficiently compared to the wild-type E. coli. As a basic strain, this acetic acid resistance capability was also improved. And then we performed another experiment to maximize the biomass of this E. coli. To maximize the biomass of E. coli, we should think about acetate excretion, because acetate may divert the carbon flow from biomass formation to the side carbon flow. And we analysis the metabolic pathway of this E. coli. We found there's two genes directly related to the acetate secretion, but the other two genes also indirectly related to the acetate production. So we constructed a series of mutants, which including single mutant, double mutant, and triple mutant, and the tetramutant. Analysis of this mutant indicated that the triple mutant and tetramutant can produce very less of acetic acid compared to the wild type. As you can see, the wild type can produce almost 6 grams per liter acetate after 20 hours incubation, but this triple mutant and tetramutant can produce only around 0.5 gram per liter acetic acid after 24 hours incubation. And the other two mutants can accumulate around 3.5 gram per liter acetic acid. And in this case, we also analyzed the glucose concentration in the medium. We found that the mutant has a lipid lag phase for this glucose assimilation or glucose consumption, but they are completely consumed after 24 hours incubation. So finally, we checked this biomass accumulation, and the wild type E. coli can only accumulate around biomass to OD about 4, while this tetramutant can accumulate the cell biomass up to almost 18, OD up to 18. So we further cultivate this mutant in pH-controlled conditions. As everybody knows that the pH may affect the growth and assimilation of these carbon sources. So we cultivate this mutant in a controlled condition. We found that this mutant did not accumulate much more compared to this uncontrolled condition, but only this wild type increased. Further analysis indicated maybe we adding too less glucose, because in the triple mutant and the tetramutant cases, the glucose has already been completely consumed after only about 10 hours. There's no carbon sources available. And interestingly, that at the pH-controlled cases, that acetate was reutilized after around 12 hours. It means that at first stage of cultivation, the acetate was first produced and accumulated, and at second stage, that the acetate was being used as a carbon source for the growth of bacteria. But the cell mass did not increase very much in this case. So by analyzing this metabolic pathway, we identified several important genes which might affect the cell mass accumulation. We also did some screening to find some regulators that might affect the biomass accumulation. Actually, we already found a protein which affects the biomass accumulation, but the further calculation is being done, so I cannot give more results here. So by maximizing the cell biomass, it could achieve a more about 5-fold than wild-type strains. So to improve the capability of E. coli for the industrialization and for this synthetic biology, not only we should consider about this cell biomass, we should also consider about induction system, because normal induction system such as IPTG is too expensive for industrialization. Temperature shift is also not suitable because in a large scale fermentation, that the temperature shift is also not suitable. Other promoter, for example, light or propionate, maybe it's also not that good for industrialization. So we developed a stress-induced promoter based on these RPOS genes. RPOS genes was actually a sigma factor that's transcribed by IPOS. IPOS regulates genes that are required for entry into survival and exist from stationary phase. It also regulates the response of bacteria to stress conditions such as osmotic shock, ethanol, nutrient limitation, low pH, and low and high temperature, as well as high cell density. Based on these properties, we set up a promoter. Based on this, on the promoter of that, we construct a GFP to monitoring the expression of this and we found that this GFP was expressed at the middle exponential phase without adding any inducer. As you can see here, it's expressed here. It's the middle exponential phase, which was very good. It's not constitutively expressed, also not expressed at very late stage. It's expressed at the middle exponential phase, and the characterization of this promoter we found is even stronger than lac promoter. As you can see, it's more stronger than lac promoter. So it's very suitable for the industrialization of E. coli. So, this developed base strain for industrialization has diminished catabolic repression. So a wide range of substrates such as aminos, xylose, sucrose can be simultaneously consumed. This strain also has maximized biomass accumulation. Of course, there's a lot of things need to be done. And this strain can resist to high acid conditions. And this strain also has a very, of course, a suitable induction system, which is suitable for large-scale fermentation without adding any inducer. And finally, we will give an example for the efficient recovery process for these strains. This example was also used PHA production because George has mentioned about these materials, which is very interesting. Actually, we also have some example for example, this strain application in the succinate acid production. But this example is more better. PHA, as George has already introduced, is a kind of biodegradable polyester accumulated by many microorganisms and unfavorable conditions. When this PHA was placed at different depths of water, a lifespan of 5 to 10 years was calculated for bottles under these conditions. But when this PHA was placed on the sediment surface, a PHA film was completely degraded within 250 days. But if we put this in the composting plant, up to 85 samples were degraded within 7 weeks. So it's very environmental-friendly materials. So it has a lot of applications. I will not go to the detail. And the metabolic pathways has already been set up in E. coli. Various PHA can be produced in this engineered E. coli. So we only using one example to explain why we could use this PHA. We use E. coli to produce PHA. This is the advantage. I will not go to the detail. I only say that it can be easier and less costly to purify polymers from recombinant E. coli from other compared to other strains. We tried this cultivation of E. coli. We found that this PHA accumulated in this strain can up to 90% cell dry weight. We make a photo, and as you can see, that the PHA are fully occupied this E. coli, and E. coli has already lived and how to say that the cell membrane has already disrupted because of it's full of PHA. For in this case, that these purification steps are very easy. Actually, when after cultivation, that this PHA is already sediment on the bottom of flask, and you can see the white type is the white is a mixture of PHA and E. coli. And of course, if you want to purify this PHA, you need a chloroform extraction. This is chloroform extraction. So, in summary, we have a diminished catabolic repression of this strain. We have maximized cell biomass. We have enabled this cheap fermentation process with suitable induction system. We have enabled the efficient recovery process for the production of PHA. And these data are obtained from these papers and some unpublished data. And I would thank my group. As you can see, they are also very happy. This work was mainly done by Kang Jin and Chen Chuan. So thank you for attention.
So we have time for a couple of questions. Please.
Do you prefer to use lac promoter for all this construction, or some other type of initiation of transcription, like invertible promoters, mechanical switches, etc., or just simply lac induction?
Yeah, actually, at the beginning of our experiment, we tried the many types of promoters. At first experiment is lac promoter, and then we tried some others. Yeah, so we finally developed our stress-induced promoter, which is based on the IPOS genes, which can regulate downstream around 10% genes.
But T7 is stronger than.
Yeah, in this case.
But then it has to be controlled, but there are a way to control by inversion.
Yeah.
Yeah. Did you, along these lines, did you ever compare your stress-inducible promoter with the T7 promoter regarding tightness and inducibility?
So I did not compare this promoter with T7, but in normal cases, T7 is not suitable for industrialization. It's suitable for protein induction. It's a very strong promoter. It can cause this protein overexpressed, and in this case, the metabolic engineering is not suitable.
Okay. Are there any other questions? Now, as this is a session for applications, how far is it from what you have achieved to product? Do you negotiate, for example, with companies for marketing your strains, or?
Not yet. We just it's only a laboratory scale, as you can see.
Mm-hmm.
All the experiment was done in a flask.
Okay.